So today we are going to discuss about the proteins. So what are the proteins? The protein are simply the important macromolecule that are present in the body, and this macromolecule is formed from the sequence of a nitrogenous basis and the nitrogenous amino acid. When the different amino acids combine in a sequence by a linkage called as a peptide linkage, this linkage is going to combine by a bond, this peptide bond is going to combine with the two amino acid or more than two amino acid to form a structure of a simple protein. And this is the important part of the living organism. Protein is very important in component of the living organism, and it has various other functions. We're going to discuss the function later on. First, we have to go discuss the classification of the protein, in which it is divided into the simple protein and the conjugated protein. So, simple protein includes the sequence of a single amino acid, sequence of a single chain of the different amino acid that are connected by a peptide linkage. What is a peptide linkage? Here you can see it is shown that when the carboxyl group C double bond O negative is combined with the amino group of the another amino acid, that is the NH3 positive group of the another amino acid combined with the carboxyl group of the first amino acid, these two amino acid with the loss of the one water molecule form the peptide bond, and this is the formation of the peptide linkage between the two amino acid, and the resulting is the formation of the protein, that is a sequence of the different amino acids. Now the conjugated protein include the one part from the protein and it combined with a non-protein group. It can be a fatty group, that is can be alcohol, can be any other macromolecule other than the protein. So when the proteinous part is combined with non-protein group of the amino acid and other structures of the macromolecule, this is formed as the conjugated protein, that is conjugated with another structure. Now, according to the type of linkage and the bonds present in between the amino acids of the protein, these are different classifications through which we can divide its structure of the protein into the primary, into secondary, into the tertiary and the quaternary structure. Starting from the primary structure, this is the primary and the simple protein, including the simple linkage of the amino acid, and there is a formation of a straight chain amino acid protein chain, in which there is only the presence of a peptide linkage with no other branching or no double bond. So the example of this type of protein is the insulin, that is very important in the regulation of the glucose metabolism of the body. Now, in the secondary structure, this primary structure is going to be branched and it combined with another bond that we call as the hydrogen bond, and other sheets it form the alpha sheet, it form the beta bands, it form the amylons, it form the other non-repetitive structures to combine with the peptide bond, that is the main bond between the protein, to form the secondary structure. Because it is not straight, it can be branched, it can be folded, and it includes the peptide bond, the hydrogen bond, and form the alpha or the beta sheets. So the example of the secondary structure in our body is the hairs. The human hair is an example of secondary structure, or you can say the spider web in the animals is also formed from the secondary structure of the protein, because it is somewhat branched or folded like structure when this start from the primary structure combining with the hydrogen bond and the peptide bond. Now coming to a tertiary structure, there is a three-dimensional structure of protein. For example, if I show you the primary structure, just take an example that the nitrogen bond is combined with the carboxyl group, then the nitrogen bond, then the carboxyl group. So this is a straight structure going on. In between, the peptide bonds are present. So this is the primary structure of the protein. In the secondary, what we can see, this structure is going to be folded in the secondary structure. Here you can see it get folded. There here's a carbon, here's a nitrogen, here's oxygen. This is the secondary structure of the protein. Here it is shown. Then in the tertiary structure, what happens? As the name suggests, the tertiary, there is a triple folding. There is a threefold of this structure that include the carbon, nitrogen and the peptide bonds between them. It's called as the tertiary structure, and this three-dimensional structure is very important, and it includes certain bonds that include the hydrogen bond, the main peptide bond, and some of the hydrophobic interaction. The hydrophobic interactions are the water insoluble interaction between the tertiary structure of protein, and the example of this tertiary protein in the human body is the lysosome, that is present in the human cell and important for the self or the autosomal digestion. Then the last is the quaternary structure, in which this three-dimensional fold is changed into a subunits. When different subunits or the similar subunits combine by different bonds, this is called as the quaternary structure, and it is arrangement of the polypeptide into a subunit structure. When this subunit having the bonds that include the hydrogen bond, the hydrophobic interactions, and the electrostatic interactions. So the example of this quaternary structure in the body is the hemoglobin, that is very important for the transport of the blood and in the formation of the iron pigment in the body. So these were the normal structures of the protein, but if there is any disease like condition, these normal foldings of the protein got damaged. How this happens? Suppose this was the primary structure going to a secondary and then there is a tertiary. If this bending is not occur, there will be an abnormal formation of protein, and this protein will going to accumulate in the blood, and if it going to accumulate in the blood or the cell, it will cause the cell death or the damage towards the body. And these proteins, the example of these proteins are amyloids and the different type of the irreversible proteins that are infectious prions and the amyloids. They are causing the damage towards the cell. Now the last thing we have left with is the functions of the protein. So firstly we have to discuss the important functions include the catalytic function. These proteins can act as a catalyst in the body, act as a catalyst in the form of enzymes. When we see that enzymes act as a biological catalyst, this is the example of the catalytic activity of the protein. Then the other function it can provide is the defense mechanism by formation of the antibodies. These antibodies are also a type of a protein molecule. Then other function is the protective function that we have discussed in the defense mechanism. By the antibodies, there is a storage of the different type of the iron, copper, and a form of ferritin, in the form of transferritin. So these are also because of the protein that is in the form of ferritin or the transferritin. Then there is a regulatory function, the regulation of the cell, the regulation of the iron, the regulation of the blood, mostly by the hemoglobin, the myoglobin. These are the examples of the protein that cause the regulation. Then the other function is the structural function. It causes the structure of a body and structure of a cell to be corrected, that formed by the collagen or the elastin. That is also another type of protein molecules. The last function is the transport, which is a major function of the protein in the body. It helps in the transport of the different nutrients, and the hemoglobin by the albumin it transport the different structures, and the by the hemoglobin transport the blood. So this was general structure and functions of the protein. I hope you will be explained well. Thank you for watching the video.